Okay, so as we discussed till Archaebacteria, we'll be starting with Eubacteria, the true bacteria that needs optimal climatic conditions to survive. So there are types of Eubacteria. One is photosynthetic bacteria and chemosynthetic bacteria. If I would use a more proper term, it would be photosynthetic autotrophs and chemosynthetic autotrophs. The one which can make their own food, but either with the help of light or with inorganic substances. Okay. So in photosynthetic bacteria or autotrophs which is there, the example they have taken is cyanobacteria, which is also called as blue-green algae. Okay. So the cyanobacteria which is there, they are photosynthetic bacteria. If they are photosynthetic, they need to have a particular pigment inside them so that the process of photosynthesis would take place. So what would be the pigment? The pigment would be chlorophyll, which is similar in case of green plants. Okay. The chlorophyll, which chlorophyll? Chlorophyll A, because there are types of chlorophyll A, B, C, and this is one of the type. These are the things which you will learn in higher classes. What are the different types of chlorophyll and what does its function? Okay. So the cyanobacteria which is there, they are colonial, they are filamentous. They're even found in freshwater marine organisms. So they form in a group and they're covered by a gelatinous sheath. So when asked about eubacteria, okay, the question can come in different ways. Eubacteria or tell us about the characteristics of cyanobacteria, all those things. So you need to know the characteristics of cyanobacteria as well. So they would be colonial, forming a group, filamentous, having a gelatinous sheath. Sheath means covering, a whole together gelatinous covering. Like that's why it has a an appearance of a very jelly-like structure because of the covering. Basically, it's kind of protective covering around them all together in a colonial form. So the cyanobacteria has shown the presence of chlorophyll A. Then there are certain photosynthetic bacteria that has the, or some bacteria there are, has the capability to fix nitrogen. Now what do you mean by fix nitrogen? Okay. Fix nitrogen means some organisms and even plants cannot take nitrogen in the form of gas, or the gaseous form cannot be assimilated by the organisms or by the plants. So that nitrogen has to be converted into different forms, nitrite, nitrate or ammonia, then only it can be used up for the production of energy. So some bacteria shows the capability to fix the nitrogen. One would be, I'll take the example of nostoc or anabena. Okay. Okay. I'll take nostoc. If you see, there are structures, heterocystitis structures. It's a barrel-shaped cell present in the microorganism in the bacteria, which does the function of fixing the nitrogen for the for the mechanisms to take place. Then the chemosynthetic bacteria. The chemosynthetic bacteria are bacteria that use inorganic substrates for they act upon inorganic substrates to get ATP, okay, for the production of ATP inside them. What is ATP? It is energy. Okay, they break down this inorganic substrate that could be nitrite or nitrate. All the substrates are broken down and the ATP production takes place for their survival. So here, both in case of photosynthetic and chemosynthetic, they are creating their own food, okay, with various mechanisms. Then comes heterotrophic bacteria. Okay? Another type of heterotrophic bacteria. They are the one that is actually abundant in the diversity of bacteria. Okay? These heterotrophic bacteria are the major members of decomposers. Now, what does the decomposers do? They feed on other dead decaying matters. Okay? So, the heterotrophic, that are dependent on other organisms for their survival. So, heterotrophic bacteria, and they contribute to a major group of decomposers. But these bacteria are used for human welfare as well. Like in your higher classes, you will be coming across a chapter called as microbes in human welfare. So the mechanism adapted by the heterotrophic bacteria to produce certain products that is being used for the welfare, for our benefits, for human benefits. For example, simple thing like the formation of curd, okay, formation of curd from milk is, sorry, the arrow should go upwards. From milk the curd is made, okay. So from milk to curd you need a bacteria, lactobacillus, okay. Even many creation of antibiotics is because of bacteria, okay. Many other products, there are n number of products in medicinal field also. Some bacteria are used. Many recombinant technology, a very common example I would use is E. coli, Escherichia coli, that is used as vector in recombinant DNA technology. It is being used, it's a very popular bacteria that is used in many recombinant DNA technology studies. So these bacteria has many benefits as well. So heterotrophic bacteria, they're used in many industries for creation of many products. And then the same heterotrophic bacteria can also be pathogenic. What do you mean by pathogenic? The ones that cause diseases. Many diseases, cholera or many bacterial infections, and in plants like citrus canker, all these are caused by bacteria. So accordingly, at one time, if the bacteria is providing you advantages, the same members of the same kingdom will have a deleterious or wrong or harmful effects on the human group. Okay, so that was heterotrophic bacteria. Like what does they do? What are the mechanism and functioning is? Then we will discuss about bacterial reproduction. Now, bacterial reproduction, which is there, I'll be writing here, okay. They normally reproduce by fission. Normal fission, asexual reproduction. But if the condition is unfavorable for the time period when they are not okay to grow, they will be forming spores. Spore formation would take place. Then as soon as the optimum climatic conditions occur, the spores would be spread and again the division would be taking place. Now, if they want to switch to sexual reproduction, or there are some bacteria that has sexual reproduction as their mechanism, that is primitive. Okay. Primitive DNA transfer. Primitive DNA. What do you mean by primitive DNA? Like there is a simple passage of DNA across the cells. Like if this is one bacteria and this is another bacteria. So there would be a space created where the genetic content which is there, it would be simply passed on to the another bacteria. Okay. Now this particular process is also called as conjugation. Conjugation. Okay. So the thing is, it does not undergo many complex mechanism and all, because you need to understand, students, the microorganisms has to divide rapidly, especially the pathogenic ones. They can't afford to have many regulatory mechanisms. What do you mean by regulatory mechanism? Okay, let me give you an example. In case of human beings, the cell cycle, okay, in cell cycle, there are many checkpoints, or many of our body functions have regulatory mechanism to ensure that whatever has been producing inside the body, that is correct, not a defective one. Because you know, a slight defect in any of the part or any functioning of the body, it may have a greater impact, a harmful impact to the body. But these bacteria or the pathogenic ones, their main agenda or process important point is to reproduce, to divide, so that if at all there are some things that are used against these pathogenic bacteria to kill them, they have to divide in such a number that even some of the members are killed, some of the species do remain. Like they don't want their species to become extinct. So if you see, if we learn health chapters and all regarding pathogens that cause disease and all, we'll see that other mechanisms like digestive mechanism, respiratory mechanism, they are not that developed as compared to reproductive system development inside the body. Some of the pathogens, their major production or major formation inside the body is reproductive system, because their main thing is to divide, to reproduce, to increase in cell number. So this sexual reproduction takes place, we know, as compared to asexual reproduction. Sexual reproduction is a bit complex, but in case of primitive DNA transfer or bacteria, it is simple genetic transfer via the cell. Yeah. So that was about bacterial reproduction. Normally they does by fission, if unfavorable climatic conditions, spore formation, if sexual reproduction, DNA transfer. Okay. Now come to mycoplasma. So mycoplasma are the smallest microorganism. So they are included in kingdom Monera, because they have similar characteristic to bacteria, but they are different at the same time. They are the smallest and they do not have cell wall. Okay, we did discuss about archaebacteria. The cell wall is there, the composition is different, and even some of the eubacteria has cell wall. But mycoplasma, which is there, they do not have cell wall, and they can grow in the absence of oxygen. They can grow in the absence of oxygen. Yeah, they can grow in the absence of oxygen, and they are also known to become pathogens of various diseases. As I had mentioned before, I will be including pics in my presentation, which will give you an idea about that what is the disease, what are the various examples of different groups, so that it becomes clear in your mind. Because if I want, I could have simply written down the examples on the board. But I know when you see it with an image, it would be more interesting and it would be clear enough to remember it. Okay, so that was all about kingdom Monera. Okay, so we'll start with kingdom Protista in the next video. Thank you.